Welcome to G Fun Facts Online. Today, we're diving into a war you can't see. A microscopic battle is raging all around us, and if we lose, modern medicine could be thrown back a century. A simple scratch could once again become a death sentence. The enemy? Not some alien invader, but our former allies, bacteria. They're evolving into superbugs resistant to our best drugs, a phenomenon called antimicrobial resistance, or AMR. It's a silent pandemic, and the stakes couldn't be higher. It wasn't always like this. Alexander Fleming's discovery of penicillin in 1928 kicked off a golden era. Antibiotics became miracle drugs, making complex surgeries, organ transplants, and cancer treatments possible. But we got complacent. We overused and misused these drugs. In both human medicine and agriculture, every time we use an antibiotic, it's like a giant, real-world evolutionary experiment. We kill off the susceptible, weak bacteria, but the strong, resistant ones survive, multiply, and pass on their defensive traits. The result? The World Health Organization calls AMR one of the top global public health threats. In 2019 alone, bacterial AMR was directly responsible for 1.27 million deaths worldwide and contributed to nearly 5 million more. We are standing at a critical juncture where the triumphs of modern medicine are at risk of being undone. So how exactly do these tiny organisms fight back? Their arsenal is surprisingly sophisticated, a testament to their evolutionary ingenuity. First, you have to understand that resistance can be either intrinsic or acquired. Intrinsic resistance is a natural, built-in property. Some bacteria just lack the part an antibiotic is designed to attack, or their outer shell is like a fortress the drug can't penetrate. But the real problem is acquired resistance, which is when a previously susceptible bacterium learns to fight back. They do this in two primary ways: through random mutations in their own DNA, or by acquiring ready-made resistance genes from other bacteria. At the molecular level, this fight comes down to four main strategies. First, there's enzymatic degradation. Think of this as the bacteria producing a weapon to destroy our weapon. The classic example is the production of enzymes called beta-lactamases. These enzymes find the core structure of some of our most important antibiotics, like penicillins, and break them apart, rendering them completely useless. The second strategy is target modification. Most antibiotics work like a key, fitting into a specific lock within the bacterial cell. To become resistant, the bacteria simply change the lock. Through tiny mutations in their genes, they alter the shape of the target protein, so the antibiotic key no longer fits and can't do its job. This is exactly how the infamous superbug MRSA, or methicillin-resistant Staphylococcus aureus, evolved. Its resistance comes from altering the protein that the antibiotic is supposed to bind to. The third pillar of resistance involves defense and evasion, reduced permeability, and efflux pumps. To work, an antibiotic has to get inside the cell, so some bacteria fight back by altering the pores in their membranes, making it much harder for the drugs to enter in the first place. Even more cunningly, many bacteria are equipped with efflux pumps. These are tiny transport proteins that act like bouncers at a club door, actively grabbing any antibiotic molecules that get inside and pumping them right back out, before they can cause any harm. These pumps are often not very specific, meaning they can recognize and expel a wide range of different antibiotics, which is a major reason we see multi-drug-resistant bugs. The fourth strategy is biofilm formation. Bacteria are social organisms. They often band together to form complex, slimy communities called biofilms. You'll find these on medical devices like catheters and implants, but also on human tissues. Inside a biofilm, the bacteria are encased in a protective matrix of sugars and proteins. This goo acts as a physical shield, preventing antibiotics from even reaching the bacteria inside. Bacteria living in a biofilm can be up to 1,000 times more resistant to antibiotics than their free-living counterparts. But what makes this threat truly terrifying is the incredible speed at which resistance can spread. This is largely thanks to a process called horizontal gene transfer, or HGT. You can think of it as bacteria having their own high-speed internet for sharing survival cheat codes. This allows resistance to move rapidly between different species of bacteria. There are three main ways this happens. The first is conjugation, where two bacteria can physically connect and transfer genetic material, usually in the form of small, circular DNA molecules called plasmids. A single plasmid can carry resistance genes for multiple different antibiotics, instantly turning a susceptible bacterium into a multi-drug-resistant superbug. The second is transformation. This is where bacteria are able to slurp up naked DNA from their environment, which might have been released from other bacteria that have died. If this free-floating DNA contains a resistance gene, the recipient can incorporate it into its own genome. And the third is transduction. In this process, viruses that infect bacteria, known as bacteriophages, can accidentally pick up a resistance gene from one bacterium and then inject it into the next one it infects. Horizontal gene transfer allows for an evolutionary pace that is far faster than what could be achieved through random mutations alone. It's the key reason we're facing bacteria that are resistant to nearly every drug we have. Faced with this incredible microbial ingenuity, you might think we're doomed. But the scientific community is fighting back with a wave of innovation. The old golden era may be over, but a new era of creative, multi-pronged approaches is dawning. First, we are trying to replenish our dwindling antibiotic arsenal. This includes tweaking and improving existing classes of antibiotics to overcome bacterial defenses. But more excitingly, researchers are hunting for completely new targets in bacteria. For example, instead of just trying to kill them, some potential drugs aim to disrupt how bacteria communicate with each other, a process called quorum sensing. If we can jam their signals, we can stop them from launching a coordinated attack. We're also turning to artificial intelligence, which can screen billions of chemical compounds on a computer to identify potential new antibiotic candidates at a speed unimaginable just a decade ago. But we're also looking beyond conventional antibiotics entirely. One of the most exciting comebacks in medicine is phage therapy. This approach uses bacteriophages, those natural predators of bacteria, as our allies. Phages are highly specific. They target only certain strains of bacteria, meaning they can kill pathogens without harming the beneficial bacteria in our microbiome. Another promising avenue is antimicrobial peptides, or AMPs. These are molecules found in almost all life, part of the innate immune system. They often work by physically disrupting the bacterial cell membrane, a mechanism that's thought to be less likely to generate resistance. We're also designing highly specific monoclonal antibodies that can target and neutralize bacteria or the toxins they produce, essentially giving our own immune system a supercharged boost. And then there are antivirulent strategies. Instead of killing the bacteria, these drugs aim to disarm them. They block the toxins and other weapons bacteria use to cause disease, which reduces the damage and allows our immune system to clear the infection. And of course, prevention is always better than a cure, so developing new vaccines against major resistant pathogens is a global health priority. Perhaps the most futuristic weapon being developed is the gene editing tool CRISPR-Cas9. Think of it as a programmable genetic scalpel. Scientists are designing CRISPR systems that can be sent into bacteria to specifically find and cut the DNA of resistance genes, effectively resensitizing them to our old antibiotics. The system can even be programmed to target essential genes in the pathogenic bacteria, killing them with surgical precision while leaving beneficial microbes unharmed. While it's still in early stages for this application, CRISPR holds immense promise as a highly specific and programmable tool to combat superbugs. Of course, this race has major hurdles. One of the biggest is economic. Developing a new drug is incredibly expensive, and for antibiotics, the return on investment is terrible. To preserve their effectiveness, new antibiotics are used as sparingly as possible, which means low sales. This paradox has led many large pharmaceutical companies to exit the field entirely, leaving the development pipeline dangerously thin. But the future of this fight will likely be defined by a shift towards more precise and personalized medicine, coupled with global cooperation. A crucial element is the development of rapid diagnostics. Imagine going to the doctor with an infection, and within minutes, a test can tell you not only which bacteria you have, but also its exact antibiotic resistance profile. This would eliminate the guesswork that leads to the overuse of broad-spectrum antibiotics. We are also entering an era of personalized infectious disease medicine. This involves tailoring treatment to both the patient and the pathogen. By using AI and analyzing a patient's own genetic data, we can better predict which drug and which dose will be most effective. Ultimately, no single country can win this arms race alone. It requires a global, coordinated effort under a One Health approach, recognizing that the health of humans, animals, and the environment are all interconnected. It requires robust antibiotic stewardship programs in hospitals and on farms to promote responsible use. The arms race against superbugs is a marathon, not a sprint. It will require sustained investment, innovation, and a global commitment to work together. The challenges are immense, but so is our capacity for ingenuity. By continuing to develop new weapons and refine our strategies, we can turn the tide in this critical battle and ensure our life-saving medicines remain effective for generations to come. Thanks for listening to Fun Facts Online.